This video focuses on the phases and importance of mitosis and meiosis. Mitosis is a process of nuclear division in eukaryotic cells. Eukaryotes depend on mitosis for their development from a fertilized egg and for growth and repair. Mitosis is important because it conserves chromosome number by distributing replicated chromosomes equally to each of the daughter nuclei. At the end of mitosis, the resulting daughter cells are each diploid and have 46 chromosomes just like the parent cell. The first stage of mitosis is G2 of interphase. The chromosomes have been duplicated but are not condensed. The nuclear membrane bounds to the nucleus. The centrosomes replicate to form two centrosomes. Each centrosome features two centrioles. The second stage of mitosis is prophase. The chromosomes are tightly coiled with sister chromatids joined together at their centromeres. The mitotic spindles begin to form and the nucleoli disappears. During prometaphase, the nuclear envelope fragments and microtubules from the spindle interact with the condensed chromosomes. Each of the two chromatids of a chromosome has a kinetochore, a specialized protein structure located at the centromere, which jerks the chromosome. During metaphase, the centromeres are now at opposite poles of the cell. The spindle fibers push the sister chromatids until they are all arranged at the metaphase plate, an imaginary plane equidistant from the poles. During anaphase, the centromeres divide, separating the sister chromatids. Each chromatid becomes a full-fledged chromosome and is pulled towards the pole to which it is attached to by the spindle fibers. By the end of anaphase, the two ends of the cell have equivalent collections of chromosomes. In telophase, two daughter nuclei form in the cell. The nucleoli reappears and the chromosomes become less condensed. Cytokinesis, the division of the cytoplasm, is well underway by late telophase. Cytokinesis involves the formation of a cleavage furrow in animal cells, which pinches the cell in two. Plant cells undergo mitosis as well. The one major contrast between plant and animal cells during mitosis is cytokinesis. In plant cells, vesicles derive from the Golgi apparatus, producing a cell plate at the middle of the cell, which will divide the cytoplasm in two.
Meiosis is a type of cell division for sexually reproducing organisms consisting of two rounds of cell division, but only one round of DNA replication. It results in cells with 23 chromosomes, half the number of a set as the original cell. This is important because it creates genetic variation in each generation. Meiosis I, the first round in the cell cycle, separates the homologous chromosomes. During prophase I, crossing over occurs where DNA molecules in non-sister chromatids break at corresponding places and then rejoin at the other chromatid. Each chromosome becomes a tetrad, or a group of 4 chromatids. Each tetrad has a chiasmatic site in which the chromatids have crossed and segments have been traded. At metaphase I, the tetrads are all arranged at the metaphase plate, with one chromosome facing each pole. Microtubules from one pole are attached to the kinetochore of one chromosome of each tetrad. In anaphase I, the homologous chromosomes separate. One chromosome moves towards each pole, guided by the spindle apparatus. Sister chromatids remain attached at the centromere and move as a single unit towards the pole. In telophase I, movement of homologous chromosomes continue until there is a haplid set at each pole. Cytokinesis usually occurs simultaneously with telophase I, forming two haploid daughter cells. Meiosis II is very similar to mitosis. Here, the sister chromatids separate. During prophase II, a spindle apparatus forms and attaches to the kinetochores of each of the sister chromatid. At metaphase II, the sister chromatids are arranged at the metaphase plate. The kinetochores of the sister chromatids attach to microtubules extending from one opposite pole. At anaphase II, the centromeres of the sister chromatids separate and two newly individual chromosomes travel towards the opposite poles. In telophase II, the chromosomes arrive at opposite poles. Nuclei from around the chromosomes and cytokinesis separates the cytoplasm. At the end of meiosis, the sister chromatids separate, resulting in 4 haploid daughter cells containing unreplicated chromosomes. Mitosis and meiosis have several key differences. The chromosome number is reduced from diploid to haploid in meiosis, but is conserved in mitosis. Mitosis produces daughter cells that are genetically identical to the parent and to each other, while meiosis produces cells that are genetically distinct from the parent cell.